told you it was going to be important. And here's a way that it's become important commercially. And that is if you understand how bacteria replicate DNA, transcribe DNA, and translate DNA, you can take advantage of it and make something. So the plasmid is going to have a few important features. First of all, it has to be able to replicate. Second of all, the gene that we are interested in has to be under a promoter that works in bacteria. So if you remember, we learned about promoters that had a -10 and a -35, and that would be great. But typically people don't just put their gene under a great promoter. The reason is, frequently, if you express a foreign protein in bacteria, they don't really like it. It goes anywhere from slowing them down to killing them. Okay? By the way, one of the ones that Dr. Furky mentioned called lactoferrin, I know from personal experience kills bacteria dead. In fact, that's its purpose. So, if you wanted to make something like that, you want to put it under a promoter that is regulated. We spent weeks learning about gene regulation. So in fact, historically, some of the first plasmids that were made to express proteins used some of the promoters we studied, such as the lac promoter and the trip promoter. However, these days that's very seldomly used, and I'll try to tell you in the next 10 minutes why that is. But why use a regulated promoter? The reason is, let's say I want to make something that the bacteria really don't like. What I want to do is put it in the bacteria, grow the cells. And what I've shown here is an example of how bacterial cells grow. If I, which I did this morning, inoculate some cells, actually my student did it because she gets in earlier, inoculated the cells. In about 2 to 3 hours, they just kind of sit there and don't do much while they're waking up. Then they start growing rapidly. And one of the things I told you guys is bacteria can divide every 20 minutes when they're really happy. And that's what's happening here. For another 2, 3, 4 hours, depending on the media and the cells, they will grow like mad. What you want to do is get them really happy, growing really well, and then turn on expression of your gene. So if you get all these happily growing cells, then you turn on expression of your gene, even if it makes them sick, you've got all these happy cells and they'll make your protein. And even if they stop growing, you've got a lot of happy cells. Then you harvest the cells and get your protein. Okay, so that's the general strategy. I want to talk about a more modern approach, which is almost always done today. And that is to take advantage of a bacteriophage and its promoter. It's a phage we haven't ever talked about before, called T7. All you have to know about T7 is that it has an RNA polymerase that only has one subunit, which is different than any of the ones we learned about, and that its promoter looks nothing like E. coli's. So those two features make it useful because I can put my gene into a plasmid that puts the gene following the T7 promoter, and as long as I just put it in E. coli, there will be absolutely no expression. So no matter how toxic the protein is, it won't be expressed because normal E. coli doesn't have T7 RNA polymerase. So I do all my cloning, manipulation, make my plasmid, then I put it into a special bacterial cell that has T7 RNA polymerase, which itself is expressed from our lac promoter. You've got to think back a few weeks to the lac promoter. If you remember, it's not turned on unless the cells have lactose. However, typically upstairs we never use lactose. We use an artificial inducer, which I believe I told you about, called IPTG, that I told you didn't have to remember what it was, other than it binds to the lac repressor and causes it to fall off the DNA. So what do? We put the cells in, we start them up in the morning. About an hour ago, I took the cells, I added IPTG. I'm going to let them grow for a while, and then I'm going to harvest it. And I'm going to hopefully get the protein of interest. So, this shows what's going on in the especially E. coli genome. The T7 RNA polymerase is under the lac promoter. I add IPDG. I make T7 RNA polymerase. It works in trans, floats around, finds my plasmid, transcribes my gene. What else do I need if I want the protein? I need a ribosome binding site. Okay? Eukaryotes don't use the same sort of translation initiation as bacteria, so I have to be careful and make sure I have a good ribosome binding site also. Okay, so let's say, for fun and profit, we wanted to clone the human growth hormone. Let's say we wanted our football team to get bigger and stronger and win. So, we are going to clone the human growth hormone, and we're going to put it into their food. We're not going to tell them because this is strictly forbidden by the NCQA. Right? And in fact, if you were to follow up this link, which I put on here, it'll tell you all the bad things about doing this. But we don't care. All right, so what do we need if I want to do this? What's the first thing I need? The gene. Okay, so I need to know the sequence of the human growth hormone gene. And I'm a little short on time, so I'm not going to actually go to this website. I'm just going to tell you there's a website that the government runs, which has the entire human genome and many, many other genomes. And if you were to click this, you can find out anything you want to know about human growth hormone. Okay, so I did that, and I found the gene. Now I have to be a little careful here. Something else we learned about. Do I want the chromosomal copy of the gene? Someone said no, why not?
Because it gets a lot of modifications.
Specifically, there's one that's really important. What might that be? Now, polyiotail's here, by the way. Splicing. Do bacteria do splicing? No. So if I put the chromosomal gene in there, I'll get more or less nothing. So I looked up the sequence of the messenger RNA, okay, which was created by a cDNA clone, which means you copied the messenger RNA and determined its sequence. Okay, and so when I look this up, it also gives me the sequence of the protein. So I'm able to say, here's the start codon. Again, this shows the coding strand. It's the sequence of the messenger RNA, which is equivalent to the coding strand of the DNA. So here I have the start codon, here I have the stop codon. This is the poly-A tail, by the way, which I don't care about in E. coli. I only want the coding sequence. So how am I going to clone this? What's the easiest way that you've recently learned about if I want to make more of this DNA so I can clone it? PCR. Okay? So I need to design some primers. And I hope that you guys could do this. So let's take a real quick look at it. The first one is, here's the start codon. So I'm going to make an oligonucleotide primer that includes the sequence from the start codon and a bit downstream. This, by the way, is 5' to 3', because that's how we always write DNA unless we label it otherwise. In addition to that, I'm going to put a little bit of extra sequence on the 5' end of my primer to create a restriction site. See how that works in a second. The other end is a little more tricky. Here's the stop codon. I want a primer downstream of the stop codon. But if I made the sequence shown here, C, 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 G, 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 etc., and did PCR, I can tell you also from personal experience, you would get absolutely nothing. Why is that? both primers are going the same way. So what do I need here? You can either think or you could read my slide. I need the reverse complement. We've been talking about this since almost day one. You need to be able to speak in the language of DNA. So if I want the reverse complement of this sequence, I hope you can all do that. I did it here. All right, so here's the sequence, and I've made, here it goes C, 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 G, G, G. If you look here, I have G, 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 C, C, C. So I have the reverse complement. I also start, just like I did up there, some extra sequence on the end. So, if I do reverse transcription PCR, I take my RNA, I copy it by reverse transcription, and then I do PCR, which you guys learned about recently, so I'm not going to go through it again. But when I do that, see these little red green slits? Those are my extra bits on here, which are going to create my restriction endonuclease recognition sites, which I'm then going to take my special expression plasmid, and you'll notice one here is called NDE1, and another one down here is called EQR1. And my piece of DNA here had an NDE1 and an EQR1 site, so I'm going to insert it there. I'm going to stick it into my plasmid, just like this, and I'm going to grow up and express the protein. Now, really quickly, just for fun, last thing I'll do, I'm curious whether anybody else has ever thought of this idea. Because I just showed you that if you learned all the things that we've done in class, you should be able to do that. So, let's see. Why are you giving me all that? Human growth hormone for sale. If you do this, this one doesn't show. When I did it upstairs on mine, you will find there's 3.6 million hits. So unfortunately, somebody already thought of this before us. You guys, as you go forward, need to think up better and more interesting and more profitable things to come in. If you have not picked up your